I should talk about genotype histophenotype correlations in congenital hyperinsulinism. And as you know, it's a highly heterogeneous disorder with a diffuse form and a focal form, where the focal form is mainly due to small endocrine lesions. But these two forms together constitute the majority of all the forms, but we have a small group that we call collectively as atypical histology. If you look into how we received the data for this study, as Henrik said, we had 188 patients coming in, performing surgery on 75. Some had insulinoma going out. Some had what we call classical forms of Chi, and only 9 actually was introduced into this type of atypical forms. If you look into how the Doppler PET-CT scan actually shows the pancreases from these patients, they actually more or less resemble a normal pancreas. Whereas if you look into the histology, here you have an overview of the histological samples, but you can see in some of these samples you have very large islets, whereas in others you have very small islets, and it's in the same patients. If you look into the morphometric measurements of this, you can see that the islets of these huge islets, they are more or less 3 times larger than the islets of the smaller islets in the same sample. And if you look into the genetics here, you'll see that some of these patients only have mutations residing in the pancreas, whereas others only have in the islets themselves. And this is really for the HK1, for the HK1 mutation that has previously been described by Flanagan and Wakeling, but we see it here in the islets only. So this is actually the first time to show mutations that are only residing in the islets itself in the HK1 gene. This has previously been shown for the ABCC8 and JCK11. So it's one of the reasons why I was talking about isolating these single cells and exosomes and stuff like that. Okay, thank you very much.